Thank you so much for inviting me for this session, and thank you, Johnson & Johnson's, for this invitation. Amivantamab seems to be the new kid on the block, and it's written all over the wall. We'll be talking about Chrysalis here because that's what I can talk, and of course the comparison between Chrysalis and the real world data, but we all saw the Papillon study, the Mariposas present, and it's Amivantamab all the way. So I think it's about time we realize the presence, and this is a smaller presence that we are going to talk about today, but we all know the larger presence that is about to come, which I can't talk today. So when we talk about EGFRs, and our world in India as a lung cancer expert is full of EGFRs, and that's the great thing, because we can treat half of our adenocarcinomas, lung adenocarcinomas, with oral therapies, and that's a very Indian thing, that's a Asian thing, and that's different from what the West talks about. But EGFRs have now been understood to be of different kinds, and we all know EGFR exon 20 insertion mutations. I think by now we have all understood that this is an alteration that doesn't really respond to a routine TKI. And identified this alteration, and you can see the kinds, as a mischief maker. And I've always said this, you put up an EGFR exon 20 and you expect the patient to respond to the routine TKI ain't happening. Then came all the different permutation and combinations that we would practice before Amivantamab came in. And a lot of us have given different kinds of combinations to try to make that pathway behave. And the struggles involved with that, and the stories with EGFR TKI and cetuximab and EGFR TKI and panitumumab, and trying to make that combination kick in and really do your bit. And so many times we have fallen our face, and there are some times when there are some anecdotal responses reported. As we talk about exon 20, this pie chart is demonstrative of us needing to understand which exon 20, and there are so many times that the patient presents, and I'm talking about with a molecular expert, this is the patient with exon 20, and then I get asked which one. And this slide demonstrates that we need to understand the differences between the exon 20s, and these are the two most commonest ones. And if you think I memorized all of them, that won't happen, right? So what I do remember is which ones are, is it the common one? Then I know that this is the drug to give. So the way I have remembered it is 67, 68, 69, 70. And between the two of them, these four are covered. And that's what I tend to remember, just to make it work for me. And between that, these two, you're actually covering most of the ones that can be targeted. So what does exon 20 insertion do? It brings on a unique biology, and it's distinct, it's different from the common EGFR mutations. If you look at the common EGFR mutations and what it does, and the interaction with the TKIs, the exon 20 insertion actually brings about a conformational change in the EGFR TKI binding site, and it just doesn't fit in anymore, and reduces the size of the drug binding pocket and the affinity for TKIs. And in the yester months and years, the thought was that if you had cetuximab come in, that conformational change would happen. And so many of us had used the combination and it didn't work really. And here you have a drug that makes that happen, but this is how the mechanism is. It brings about, the exon 20 insertion brings about a conformational change, and the TKI which used to fit no longer fits in that area anymore. So we have the stories of frontline combination, the second line combination, and platinum-based therapies in frontline and second line setting that have actually behaved in an inferior way for this particular alteration. And bottom line, there is no standard of care for this particular molecular finding. And if you look at this study of over 3,000 patients with advanced EGFR-mutated NSCLC and exon 20 insertions, compared with common EGFR mutation, and you see how in this real-world setting the mutation, the alteration behave. Patients with exon 20 insertion had poorer prognosis and 75% increased risk of death. So this was no longer realized to be the golden EGFR story. These were the patients that fell out of that, the golden, the rosy story that we paint of EGFR mutated patients. And so these were the misbehaving alterations recognized. And then you have over 2,000, almost 3,000 patients again in this study, where in the real world progression-free survival was significantly improved for the routine EGFRs versus exon 20 insertions. And the writing on the wall, real poor prognosis, 169% increased risk of progression or death with routine EGFR TKIs. And here comes the breakthrough with Amivantamab. And so Amivantamab was developed to target both EGFR and MET mutations, and you have this bispecific antibody, and the Hindu mythology can truly explain that. You have an EGFR binding site, you have the MET binding site, and you do the works much better. These are the two most required pathways for this, in the setting where the drug was developed. And so Amivantamab is a fully humanized EGFR MET bispecific antibody and targets the activating and resistance pathway for EGFR mutation and MET mutations and amplifications. And then you see how this drug was, the realization that the Amivantamab binds extracellularly, so it's not affected by the co-mutations in the EGFR TKI binding sites. By targeting the activating and the resistance EGFR mutation and MET mutation and amplification, Amivantamab addressed two major mechanisms of standard of care resistance. And then different features of the drug, where you have immune cell directing activity, you have the receptor degradation, you have the inhibition of ligand binding, and all three bring together the final outcomes that you see with Amivantamab. And in this, you see how the immune cell directing activity, the ADCC, antibody-mediated, antibody-directed cellular cytotoxicity that happens when Amivantamab as a molecule comes in contact with the receptors of the tumor cells and leads to activation of the immune effector cells. You can see in the first diagram. And then you also see the trogocytosis and the tumor cell lysis that is brought about by the ADCC mechanism. And then, of course, the receptor degradation that Amivantamab, by binding to the EGFR and the MET receptor, leads to the receptor inactivation. The third way of working is inhibition of the ligand binding site, and you can see both for the EGFR and MET, it achieves its goal. Dosing and administration, we all know that it is divided in day 1 and day 2, the week 1, and the first month is week 1, 2, 3, 4, and then forward in the subsequent weeks, it could then be administered in a monthly manner. So for the less than 80 kilograms body weight patient, the dose is lesser, 1,050. Above 80 kilograms, you have 1,400 milligrams. And The pre-medications are most important because of what we realize now is the infusion reactions that can happen in these patients. And so the commonest time period where it happens is the first day, and we have to be very careful with the pre-medications that we give on. So it's day 1 and day 2 of administration of the antihistaminic, antipyretic, the glucocorticoids, and very carefully administer on the day 1, day 2, the first week, and then of course the week 2 and week 3 and week 4. And the highest toxicity or infusion reaction is observed on the day 1 of the administration. We have to be aware of that. Chrysalis study is in the cohort B. After the dose was established in the part 1, you have the cohort B where the safety data is now available, and you have enlisted safety with infusion reactions and rash as being the commonest adverse reactions that we see with Amivantamab. And those of us who are privy to other drugs that work in this particular pathway, we are aware that there could be infusion reactions, and it is rare with the other EGFR inhibitors, but it is very commonly seen with this particular drug. As long as we are aware and we know how to manage, this is very safe, and the drug discontinuations are actually minimal because of the toxicity seen with infusion reactions. So if you see here, treatment-related grade 3 adverse events was reported in 18 patients in the Chrysalis study, which is 16%. Rash was 4%, infusion reaction neutropenia was 3%. And serious adverse events were again with infusion reaction and diarrhea. Dose reductions were seen in 13% in total, and discontinuation was seen in 4%. Out of that, the rash and the infusion reaction was responsible for dose discontinuation in 1.8% of patients. And here is the response rates. And you see the CR and the PR rates are listed here. And the clinical benefit ratio goes up to 74%. And the response over time is also outlined here with a median progression-free survival of 8.3 months and the overall survival, median overall survival of 22.8 months. And you see efficacy across all the exon 20 insertions. I think I'm all out of time, but I've already mentioned the safety results, and I'll scoot over to the last section where we look at the progression-free survival and the overall survival via Chrysalis, but we are all aware of the studies that have been presented, the Papillon and the Mariposa, and the results of those studies. I cannot be quoting those right now, but long-term clinical benefit of Amivantamab was seen in 48 out of the total number of patients, which is 42%, and the clinical benefit was most clear in the patients who had received above 12 cycles of this drug. The best results were seen in patients who had good performance status. So ECOG-PS of 0 was synonymous with the best responses. And we have the real-world data where you see Chrysalis performed much better than the real-world data for this particular alteration treated with other drugs. Significant benefit in overall survival has also been demonstrated. And finally, we have the new drug, which we all need to understand if we haven't understood yet. The specific that we are talking about today, including the Chrysalis and the real-world data, is about exon 20 insertion. We have more data that has already been presented and will be privy to us, must be privy to us by now, more discussions to come. But through the Papillon study, and in the regular EGFR patients where, routine EGFR mutations where we've already exposed our patients to osimertinib, we see the Mariposa study. So thank you. I'll leave you for some food for thought. And those of you who can't avoid Amivantamab anymore. Thank you so much. Thanks for the invitation.